Hello, gentlemen. As I said on class on Friday, I was going to give you some recorded lectures. This is going to be the first of two lectures that you're going to be listening to before class next Friday. This lecture, we're going to be doing two things. We're going to be reviewing the biotech methods that we discussed on Friday, so we can get some review going on there. We'd also like to complete this lecture by discussing two new methods called the Southern blot analysis, as well as DNA sequencing, or simply just sequencing the nucleotide sequence of a stretch of DNA. The other lecture that you're going to be exploring before Friday is a lecture on the specific application that we're going to be testing in our laboratory investigation on Friday, which is known as transformation. So be aware and stay tuned for that lecture as well. All right, to begin our review, let's just make sure that we're understanding that we're really talking a lot about DNA and the applications of gene expression. One major tool in manipulating gene expression is this thing that we're going to be calling a plasmid, and specifically a recombinant plasmid, which allows us to insert DNA into a plasmid and then insert that plasmid into some kind of bacterium. All right, this slide, as we saw on Friday, shows us the method of biotechnology known as DNA cloning. We're going to clone DNA when we want our gene of interest. All right, we're going to cut it out of the organism with a restriction enzyme. We're going to insert it into a plasmid. Here's our plasmid. And that plasmid is special because it has something that will allow us to understand which bacteria have taken the plasmid up. This special gene is known as ampicillin resistance. We're going to see something interesting there in our lab on Friday. So once we've inserted this gene, we've put it together and glued it together with ligase. So here's ligase over here. We're going to put it back into the bacteria by a method known as transformation. That's going to be our lab on Friday. Then we'll grow it on a nutrient agar plate. Okay, and so ultimately bacteria just grow and duplicate, and you're going to see a bunch of different colonies on this agar plate. Some of the colonies will have the gene of interest. Some of them will have other genes or other DNA inserted into it. All right, this is a nutrient agar plate on which bacteria has been growing. Each of these dots represents a bacterial colony, which means that it's literally millions of identical bacterial cells. Identical meaning that if it's taken up a plasmid, it's taken up the plasmid, and it has the identical fragment of DNA inserted into it. Again, some of those plasmids have taken up our gene of interest. Others have taken up other DNA that's not our gene of interest. And now our job is to figure out and to detect which colonies, maybe this colony is the one that will have our gene of interest, maybe this one will. We don't know. We're going to figure it out by screening with a nucleic acid probe. But even though not every bacterial colony has taken up our gene of interest, all of the colonies as a whole represent what we're going to be calling a genomic library. That means that each colony has taken up a different or at least many of the colonies have taken up different stretches of DNA from the original organism, such that we can use each colony as a special location along that genome. We can then eventually screen that library to understand which plasmids have which genetic information. So if we have our genomic library, whether that's on an agar plate or whether that's on a multi-well plate, we can then apply to all of the colonies a nucleic acid probe, a probe that will hopefully either be chemiluminescent or radioactive. Either way, the probe will attach to the complementary sequence of the gene of interest, so that when we overlay, whether it's a photographic film or maybe it's UV light, we can identify which specific colonies have our interested or our gene of interest. Once we understand which colonies among the library have taken up our gene of interest, then we can select those colonies, cut up those colonies' genomes with restriction enzymes, all right, with our restriction enzymes, so that we have a bunch of fragments. With those fragments, we can put those fragments inside a well of a gel. The gel would be something like a polyacrylamide or some kind of polysaccharide gel. And then we can apply an electric current that will separate all of those fragments on the basis of their size. And so now what we've done is we've taken maybe a genome or a section of DNA from an organism that we know has our gene of interest. We've split that genome into fragments, and now we can again apply a new probe. All right, we're going to apply a probe to figure out where along this DNA, specifically, our gene of interest lies. That method of doing so is what we're now going to call a southern blot. All right, now we're getting to stuff that we haven't talked about on Friday. We're talking about this method called the southern blot. It's named after a guy whose last name was actually Southern, so it actually has nothing to do with the cardinal directions. However, there are other blotting techniques that are similar to this that use this that make a pun on this guy's name Southern. So there's another thing called the western blot and the northern blot. But be aware that this is a capital S for the reason that it is somebody's proper name. What a southern blot is, however, is a method by which we can take a specific sequence of DNA and again probe it to understand exactly where along that DNA we find a gene of interest. All right, the way we do it is we take our electrophoresis gel. All right, again with the restriction enzymes, we fragment DNA, we filter it on the basis of size, and we're going to get some banding patterns. Okay, here's our banding pattern. All right, now we take this gel and we apply a specific suite of tests in which we have an alkali or basic solution, we have a sponge. Here's our agarose gel, that's our yellow gel, or excuse me, that's the gray gel, the sponge is the yellow. Right above the agarose gel is this special kind of paper known as nitrocellulose paper. Nitrocellulose paper is a specific kind of paper that has a high binding affinity to things like DNA. And then you have got a stack of paper towels, and what we'll be doing is we'll be applying pressure down upon this stack of paper towels so that the DNA on the agarose gel will literally bind to the nitrocellulose paper. You'll eventually remove the nitrocellulose paper, has DNA bound directly to it. So we've actually literally transferred the DNA from the gel to the nitrocellulose. Ultimately, we're going to then add a probe, that is hybridized to our separated DNA. And that probe will tell us specifically which band has the DNA or the gene of interest that we're looking for. This is a similar diagram. You can pause to take a look at it, but I'm not going to spend too much time on it. What I do want to bring your attention to is, again, that that probe will be something like a radioactive probe or some kind of chemiluminescent probe. And so you're going to, again, apply some kind of photographic paper to exactly determine which bands have a specific the gene of interest associated with it, right? And so that's what we're looking at over here. Once we've screened our electrophoresed gel, once we've screened our DNA using the southern blot technique, now we've got a specific target sequence of DNA that we have a small amount of that we'd like to amplify. Again, we can do such amplification with the method that we talked about on Friday, known as PCR, or the polymerase chain reaction. Again, this polymerase chain reaction amplifies DNA by using some kind of complementary primers to the gene of interest of each complementary strand. We use DNA polymerase to extend those strands. And if you recall, if you take a look closely, We need to perform this technique at least 2 or 3 times so that we start to begin to generate the small shortened strands and no strands that have extended DNA associated with it. If we do this a number of times for a number of rounds, like 40 or 50 times, we're going to exponentially grow this amount of DNA, and we'll have enough DNA to work with, such that then maybe we can sequence this gene of interest. Maybe we can sequence and understand specifically what the nucleic acid sequence of the gene is. With that, let's take a look at how we can sequence DNA. Sequencing DNA is a special technique to understand specifically which nucleotides go one after another. Of course, we're talking about the nucleotides like adenine, cytosine, guanine, and thymine. And sequencing DNA, much like the polymerase chain reaction, or PCR, much like DNA replication, utilizes many of the same tools. We need a template strand. We need a primer so that DNA polymerase can attach and start polymerizing or extending that strand. We need our nucleotides, or specifically our deoxyribonucleotides. And we have in this method, we're going to add a bunch of adenines, a bunch of cytosines, a bunch of thymines, and a bunch of guanines. All right. Obviously, these are triphosphate bases, and so eventually you're going to get rid of 2 of the phosphates by cleaving that bond right there. But the special part of DNA sequencing is this last tool that we're going to use, or this last part, where we're going to call them the di-deoxyribonucleotides. When I say di-deoxyribo, di meaning 2, deoxyribo meaning the ribose has oxygen removed. So in this case, we're taking 2 oxygens and removing them. So our deoxyribonucleic acid specifically still has 1 hydroxyl group. The di-deoxyribo has also taken away that hydroxyl group. What's special about this now is the hydroxyl group specifically is what allows one nucleic acid to bind to another nucleic acid, or one nucleotide to bind to another nucleotide. So what happens is if you incorporate a di-deoxyribonucleotide, it will ultimately stop the polymerase, or the polymerization of that strand of DNA. Ultimately, another nucleotide cannot bind to that di-deoxyribonucleotide. Okay, so it stops the polymerization process. More importantly, that di-deoxyribonucleotide also has a fluorescent tag. That's going to be important for us in a second. So let's take a look at how this whole process works. So in order to sequence our DNA strand, we've got the solution that has our deoxyribonucleotides, all of them and a bunch of them. We also have our di-deoxyribonucleotides that are fluorescently tagged. And then we have all of the machinery necessary in order to polymerize a strand of DNA. So what happens is, in this solution, we've got our DNA polymerase and with our primer, and what's going to happen is ultimately the polymerization reaction is going to occur, and it's going to polymerize DNA. But every time a di-deoxyribonucleotide is attached to the growing strand, polymerization will stop because a new nucleotide cannot bind following the di-deoxyribonucleotide. This reaction happens so frequently and so often and randomly, such that one of every possible sequence of DNA is created. What I mean by this, of course, is that fragments of DNA are created, much like fragments of DNA in a gel electrophoresis. However, so many fragments are created, and in fact, every combination of fragment or every length of fragment is created, and these fragments are different simply by 1 nucleotide. So this fragment is identical to this fragment except for this last nucleotide. And that last nucleotide is a fluorescently bound nucleotide. And so we've got a bunch of different fragments. What we'll then use these fragments to do is another electrophoresis. We're going to be inserting this electrophoresis into this, not a gel, but a capillary tube, a very small and a very thin tube. It's thin so that a laser can pass through it. And once this laser passes through a particular portion of the tube, it will then be able to detect the kind of fluorescent tag. That is to say, if the tag gives off a purple wavelength of light, it will indicate a purple wavelength of light. And that purple wavelength of light will always be associated, for example, with adenine. A green wavelength of light would always be associated with guanine. A yellow wavelength of light would always be associated with thymine. And in this way, if we can detect the different wavelengths of light and the sequence of those wavelengths, then we can understand specifically what the sequence of DNA nucleotides are. And as a result, we have then sequenced our DNA. What we see here is an example of a DNA sequencing result. You can see that there are many bases, many nucleotides, and each nucleotide is associated with a specific color. So we see a sequence of colors, and those sequence of colors suggests a sequence of nucleotides, and therefore our DNA sequence. All right, this concludes our lecture to actually conclude our lecture from Friday. Stay tuned again for another lecture talking specifically about transformation and the kinds of sterile technique that you will be required to perform on Friday in lab. All right, I'll see you guys later.